dates back to Temple University, where he received his MD from and did a lot of his training. I think there was a year in between where you were at Mass General.
San Diego, Mass General, back to Philly, Temple. I'm a gypsy.
I was here. Anyway, Doc has done a lot of work on related to heart failure and I remember tumor necrosis in cardiac tissue. And he'll tell us, and as you know, heart failure involves a lot of remodeling of the tissue that affects everything, including electrophysiology. So we'll hear from Doug about remodeling in the context of the heart.
So, Yoram, thanks for the invitation to speak. The work in the lab really for the last 15 years has focused in on the role of inflammation in heart failure. And coming to WashU, we've sort of taken a little different spin on how we want to approach things, and I'll touch on that at the end. But what I want to do really over the first part of the talk is to introduce you a little bit to what I think drives heart failure. And then the middle part of the talk will be basically studies that have been already published that sort of parse out this whole process of cardiac remodeling. And at the end, I'll share with you some new unpublished data and sort of an approach we want to take here in collaboration with Charles Gu, using sort of a systematic approach, biosystems approach to cardiac remodeling, this process known as cardiac recovery. And Tom, if I mess up, you're going to bail me out, right?
Hopefully, yes.
All right. First time with a Mac, so I'm nervous. And understand the last speaker finished the whole bottle of wine. I promise you I will finish this cup of coffee, but I will walk out of here without escort. Okay, so for those of us in the heart failure mafia, most of us think that heart failure begins after a so-called initial index event produces a decline in pumping capacity of the heart. Now, the index event can be an acute myocardial infarction, it can be a sustained pressure or volume overload, it can be a genetic abnormality that disrupts contraction. It really doesn't matter. The initial index event produces a decline in pumping capacity. In response, a variety of compensatory mechanisms are activated, and the ones that we think are most important today are the renin-angiotensin system and the adrenergic system. We also think that they crosstalk with the inflammatory system. And the prevailing wisdom of these compensatory mechanisms preserve cardiovascular homeostasis for a period of months to years, during which time patients largely remain asymptomatic. So you can have an ejection fraction of 20% or depressed pump function and not know it. In fact, about 2% of the American population is walking around with asymptomatic heart failure. They don't know it. The prevailing wisdom, at least in 2009, is that when these compensatory mechanisms are excessively activated, they in turn produce secondary damage to the heart and the circulation, and that's really what drives the disease process forward, and patients undergo this transition to symptomatic heart failure, come into the hospital, and ultimately die. So this process of secondary damage largely works through cardiac remodeling, wherein the ventricles not only get bigger, but the walls thin, and the pumping capacity decreases, largely as a surrogate measure, really, of this remodeling process. So for those of us who are interested sort of in the heart failure world, this process of cardiac remodeling has become the holy grail in terms of understanding the biology, developing new strategies to prevent it. And as I'll touch on at the end, we're beginning to try to look at ways of taking these big hearts and making them smaller through a process known as myocardial recovery. So when I was a medical student, heart failure was thought to be a one-way street. You could never go back down. Today we know with a lot of the therapies we have that actually the plasticity of the myocardium is remarkable, and that these really big, baggy, dilated hearts can recover. And with it, patients improve and actually can have more normal lifespans. So obviously this is a sort of a chicken and an egg type question. All advanced heart failure, the heart gets big, baggy, and dilated. So the question is, does remodeling really cause the disease, or is it really a fellow traveler? So I'm going to try to come back to this at the end of my talk today. But one could argue that cardiac remodeling is really just an end organ response and has nothing to do with the pathophysiology. Now the reason why I think it's important is shown on this slide. So this just shows a cross-section of a ventricle, a normal ventricle with an ejection fraction of 60%, stroke volume of 100 milliliters. And you can see that as this ventricle undergoes cardiac remodeling, the ejection fraction goes down, because the way we calculate this is N diastolic volume minus N systolic volume over the N diastolic volume. So a lot of what we measure with EF really is the fact that the ventricle is getting bigger. The denominator gets bigger, so the ejection fraction goes down. But you can see here that stroke volume is largely preserved. So this remodeled ventricle here preserves stroke volume, but it does so at the expense of a number of things that can drive heart failure forward. So the first thing everybody here remembers the law of Laplace. So as the ventricle gets bigger, the wall stress increases. That's the amount of work that the ventricle does. This leads to increased afterload on the ventricle, the amount of work that it has to do to eject blood. This in turn can lead to increased oxygen consumption and episodic subendocardial ischemia. We're beginning to realize now that people with heart failure, as they get worse, they begin to basically necrose off the inner shell of their ventricle, which again leads to worsening remodeling. There's this concept of stretching myocardial tissue leads to activation of a variety of maladaptive genes. I actually started my research career stretching cardiac myocytes, and subsequent work shown by others has really demonstrated, I think, clearly that stretching myocardial tissue will activate maladaptive genes. There's this whole concept that as the ventricle gets bigger, the papillary muscles become incompetent, and the ventricle begins to leak blood backwards, and that leads to a sustained volume overload on the ventricle, which makes it worse. And there's this other concept that we've introduced, and I think is supported by the literature, that as these ventricles get bigger, all of the compensatory mechanisms that I talked about really don't work anymore. And why that's important if you wear a white jacket is that we give a lot of drugs to people, and at the end of the course, they really don't work. And so one of the stimuli or impetuses for developing novel therapies to treat cardiac remodeling is that basically all of the drugs that we have to save lives only really work in smaller ventricles. So we were really pretty proud of ourselves about sort of trying to link cardiac remodeling as a pathophysiologic cause. And I sort of presented the concept to one of the senior heart failure physicians, and Arnie Katz, who's like the historian for the Heart Failure Society. So he basically pointed out that John Bell in the mid-1700s had actually sort of come up with this, that the heart may be too big for its system is a melancholy fact. For when it becomes relaxed, it enlarges, and as it grows in size, it loses its power. Often there is formed such an accumulation of ill-oxygenated blood that suppresses the vital powers and chokes the motions of the heart. So basically the concept that bigger is not necessarily better goes back to the mid-1700s. But the guy who actually, I think, got it right in terms of the modern perception was Linsbach, who was a pathologist. And here he just states that structural dilation is the morphologic substrate for the decompensated heart, and that the cross-sectional area of muscle capable of contracting is too small for the increased volume of the ventricle. And cardiac insufficiency, i.e. heart failure, results not from overstretching of the fibers or inadequate oxygen consumption, which were the theories of the day, but basically because of strictly mechanical reasons. So this whole concept is that the big ventricle really can't contract well and sets itself up for a variety of downward spiral scenarios that lead to worsening heart failure. So what I want to do now is review with you, and I realize that there are very few MDs in the room, but just talk a little bit about all the heart failure therapies that we have today and show you that each of these therapies actually allows the heart to get better. And the reason for sort of going through this is to develop this concept of myocardial recovery, for which there is a specific molecular signature, we think. So the first paper to really do this, and this was an example of translational medicine at its finest. Mark Pfeffer's wife, Janice Pfeffer, was this wonderful scientist who did a lot of the original work with remodeling. What they did was to create heart attacks in rats. And then they gave him a drug called captopril, which blocks the renin-angiotensin system, one of those compensatory mechanisms. And they showed that it prevented the hearts from dilating in the rats. The husband, then being very clever, did the same thing in people. And I don't think we've ever really reproduced this quite as successfully, but the so-called Pfeffer models become the thing that we all try to emulate in the lab. But the bottom line here is what they did was to look at ventricular size and then look at patient outcomes. And you can see here, this is the left ventricular size, so these are smaller, these are bigger ventricles. So the people with the biggest ventricles in N diastole and also N systole actually had the worst outcomes. So again, this concept that these big, baggy ventricles don't do well. But the interesting thing is shown on this slide is what they did was they looked at then how people responded to the drug. So the people with the smaller ventricles actually responded quite well to the drug. You can see that the people who had the bigger ventricles, actually there was no response of captopril. And this goes back to my comment about the so-called neurohormonal escape, that these big baggy ventricles really don't respond to drugs anymore, and that the disease is driven largely at the end stages purely by mechanical means. So I'm going to just go through a series of studies now. We traditionally use about 2 to 3 different drugs in treating patients with heart failure. The first one of these ACE inhibitors, and this just shows a study done with radionuclide ventriculography, a way of measuring volumes. And what they did was there were 2 trials called studies on left ventricular dysfunction. One was in mild heart failure, and that was the so-called prevention trial. And another one was the so-called treatment trial, where the people were sicker. So what they did was to look at changes in end diastolic volume on drug and off drug. And you can see here, in the milder patients, the volumes are smaller compared to the sicker patients. Again, this goes to the concept that remodeling drives the disease process forward. But you can see here for the people who were randomized to receive active drug, the ventricles actually got smaller. This is, again, a signature of so-called myocardial recovery. And people lived longer, stayed out of the hospital better, had less cardiac arrhythmias, a variety of things. Similar stuff, or findings can be observed if one looks at beta blockade in left ventricular remodeling. Here they used a drug called carvedilol, blocks the beta-1, beta-2 receptor, and the alpha receptor. And you can see again that the ventricles got smaller in these people, and this was associated with about a 35% reduction in all-cause mortality. So again, the theme here is that drugs that cause the heart to get smaller, i.e. myocardial recovery, allow for improved patient outcomes. I wanted to present at least something relevant to your group, Rudy. So when you look at how biventricular pacing works in heart, this is where we have dysynchronous ventricles and we pace from the LV free wall and the septum. And you couldn't do this study today because now we know that these devices save lives, but when I was on the editorial board in Circulation back in 2002, I kind of had a fight to get this on. But what they did was to look at ventricular volumes in people who had heart failure. And when they turned the BiV pacer on, the ventricles got smaller and the amount of microregurgitation got less. Then when we turned the BiV pacer off, you can see that the ventricles forward were modeled. And today we know that the biventricular pacers are associated with decreased hospitalization and also improved survival. So if cardiac remodeling contributes to the progression of heart failure, and I haven't shown you all the data because I wanted to focus in on the science, and all current therapies favorably impact cardiac remodeling, is it possible to develop new therapeutic strategies that alter heart failure outcomes by directly preventing cardiac remodeling? So that's sort of been the theme for the lab over the past 5 to 6 years. So what I want to do is show you some work we've done with a transgenic mouse that overexpresses a molecule known as tumor necrosis factor, which is an inflammatory mediator. And the reason we got into creating a mouse was years ago we made the observation in the lab that people with heart failure expressed these inflammatory mediators in the heart. Prior to that, it had only been expressed by inflammatory cells. So what Guillermo Torre, who's now director of heart failure transplant at Methodist, did was we got non-failing tissue and tissue from ischemic cardiomyopathy and dilated cardiomyopathy, and then we just simply immunostained for TNF protein, and you can see on these cardiac myocytes that it's immunolocalized. There was gene expression for TNF, and TNF is almost never expressed in a normal heart. And then when we ground up the hearts and looked at TNF protein levels, you can see that there were really robust levels of the protein. So this, we wanted to know what the physiologic consequences of this. So what we did was to develop a transgenic mouse model that overexpressed TNF in the cardiac compartment itself. And you can see here, these are the phenotypes of the mice. And the mice that overexpress TNF undergo this process of cardiac remodeling. Again, you can see the cavity dilatation and mild wall thinning. And they develop myofibrillar array, a disarray, which is another signature of heart failure. And if you put a small malar catheter in them, they actually have contractile dysfunction. They don't develop pressure as well, and they don't relax as well. And then if you do some other cute things, like put them on treadmills, you can show that the mice can't run as far. And so we did this in collaboration with people at the Mayo Clinic. The TNF transgenic mice, they don't run as far. When you actually measure leg strength, gastroc strength, it's not because the muscles are weaker. They actually have diaphragm weakness and they develop water in the lungs, just like heart failure patients. If if you look at mortality, which is what we do in heart failure trials, you can see that the mice with the TNF transgene actually die much faster than the littermate controls. And then this is what a littermate looks like at about probably 12 to 14 weeks, and this is what the TNF transgenic looks like. The mouse with a bad hair, having a bad hair day is actually the TNF transgenic. This is actually all edema in the tissues and in the face. In heart failure, we refer to this as lean and facies. But the mice actually develop pulmonary edema just like a heart failure mouse. So we were interested in trying to understand what was driving the cardiac remodeling in the mice. And so what we did was first do what we always do in cardiology, is we take pictures of the heart. And so these are MRIs that were done in collaboration with Jacques Pichon at Dallas Southwestern. And this is the heart cross-section here, obviously. This is the RV, this is the LV. You can see that TNF transgenics initially develop a concentric hypertrophy phenotype. The wall's thick and the cavity dimensions normal. By about 12 weeks of age, they kind of recapitulate the heart failure phenotype. The cavity begins to dilate and the wall's thin. And if one just measures this with MRI, which is exquisitely sensitive, you can see that the end diastolic volumes increase about 4 to 5-fold. And if one looks at the ratio of the wall thickness to the radius of the ventricle, the so-called R to H ratio, which is a very poor man's way of looking at wall stress, but it also tips you off to adverse remodeling. You can see the R to H ratio increases. And all that means is that the cavity is dilating and that the walls are not thickening appropriately. Normally, as you grow from an infant to an adult, your R to H ratio remains invariant. So your ventricles get bigger, but the walls get thicker to match that, so that wall stress stays matched. And you can see here that the R to H ratio is changing. So we became interested in studying this. And so there are a lot of reasons why the ventricle can remodel. One of the ones that we knew from the literature was that TNF activates a variety of enzymes that exist in the matrix that dissolve collagen, so-called matrix metalloproteinases. So in collaboration with Frank Spinelli, who's a good colleague from South Carolina, what we did was to just look at the collagen content in these mice. Collagen actually keeps the heart structurally intact, and if the collagen gets blown out, you can imagine that the heart might dilate under increased pressure. So these are transmission electron micrographs. You can see the collagen myofibrils here. And in about 4 weeks of age, you can see that the myocardium becomes denuded of collagen. And if one does picroseries red staining, which is a way of looking at total collagen content, you can see that it goes down. And then interestingly, at about 12 weeks of age, the mice actually get increased fibrosis. And Wei Li, who is our Wei Li's audience has been studying this, and actually a lot of this is driven by influx of mast cells into the myocardium and crosstalk between mast cells and fibroblasts. But anyway, we wanted to kind of link this to matrix metalloproteinase activity. So again, we did gel zymography, which if you look at the zymograms here, you can see that there's increased MMP activation in the transgenic mice at about 4 weeks of age, and this tends to get turned off by about 12 weeks of age. And if one just looks at the group data, you can see the MMPs are activated and that they tend to get deactivated at about 12 weeks of age. Now, it turns out that nature has developed ways to regulate matrix metalloproteinases, and the natural inhibitors of matrix metalloproteinases are these so-called tissue inhibitors of matrix metalloproteinases, of which 4 species exist in the heart. And so what we did was just to look at the increase in, or the change in TIMP levels in the mice. And you can see that basically at about 12 weeks of age, the TIMP levels go up. And this is driven by a variety of different factors, including TGF-beta, which is upregulated in the heart. But the important thing, if one looks at the ratio of MMPs to TIMPs, which would tip you one way or the other. So if MMPs are high and TIMPs are low, you're going to degrade collagen, you're going to dilate. If MMPs are low and TIMPs are high, what you'll do is you'll become more profibrotic. And when we did that in these mice, you can see that early on the MMP activity outweighs the TIMPs, but when you get more profibrotic, the TIMP, this ratio goes down largely because the TIMPs are going up. So what we wanted to do was to prove that the MMPs were actually involved. And so we got the tissue inhibitor, we got the matrix metalloproteinase inhibitors from Procter & Gamble through our collaboration with Frank Spinelli. And we just fed the mice the MFG inhibitors for a period of about 4 weeks. And so the diluent-treated mice are shown here by the hatched bars. And so when you give diluent to the TNF transgenics, they dilate, as I showed you before. If you just inhibit the matrix metalloproteinases, you can completely prevent the cardiac dilatation, but it had really no effect on wall thickening. So just to summarize what we've talked about so far is that we think these inflammatory mediators are activated in response to tissue injury. And what they do initially is the MMPs, they'll activate MMPs, and then it turns out that MMPs will only cause dilation if there's an increase in LV pressure. But we think that this is largely responsible for the LV dilatation that you see, but it didn't explain the change in wall thickness. So at about the time we were doing these studies, there was an RFA from the NIH to go look at apoptosis and heart failure. And so what we did was just ask the question, is there apoptosis in the TNF transgenics? And again, this is the model that we're using. And the interesting thing here was that when we looked at apoptosis with a fairly specific technique called the ligase technique, you can see that there was a lot of apoptosis in the mice at 4 weeks. But the interesting thing was that the apoptosis actually increased from 4 to 12 weeks of age. Now, if you think about it, you're expressing a transgene at a constant level. So it's not that the TNF is becoming more. The question is why then would the prevalence of apoptosis increase in these mice? So we kind of set out to understand this. And before I do it, I wanted to review TNF signaling apoptosis. It's a little complex, like all things with inflammatory signaling. But there are 2 basic pathways that drive cell death through apoptotic processes. The so-called extrinsic pathway, for which the TNF got its name, tumor necrosis factor, and the so-called intrinsic or type 2 pathway, which is largely driven by the mitochondria. So TNF first came to attention because it's got a so-called death domain in it, and the death domain attracts a variety of scaffolding proteins that in and of themselves don't induce apoptosis. When they disengage from the TNF receptor and then go into the cytosol, they recruit a fast-associated death domain protein, which then activates caspase-8, which will cleave BID, which then inserts into the mitochondria and leads to dumping of cytochrome C, which initiates the apoptosis pathway. The so-called intrinsic pathway is understood less well, but it's very clear that TNF can activate this pathway, and when it does, it can lead to apoptotic cell death through a caspase-independent pathway, largely through release of endo-G and AIF, apoptosis-inducing factor, which go directly to the nucleus and cleave DNA. Or more commonly, when the mitochondria depolarize, they'll dump cytochrome C, which then activates caspase-9 and 3, which can then go lead to cleavage of DNA. So both of these pathways exist in the myocardium. Under normal conditions, TNF is actually not a bad player, but as I just showed you, sustained levels of it will lead to apoptosis, and we wanted to kind of work on the mechanism of this. We decided to interrogate the so-called intrinsic pathway first, and so what we did was just measure cytochrome C release. Again, that's when the mitochondria depolarize, they'll dump this and they activate the caspases. And you can see that it was actually a progressive release of cytochrome C into the cytosol. And this coincided nicely with the increased prevalence of apoptosis that we were seeing. We also measured caspases 3 and 9 at 12 weeks of age. And you can see that they were elevated. So we had pretty good evidence that the so-called intrinsic pathway was activated. And what we wanted to do was to try to better understand what the molecular underpinnings of that were. And we used a ribonuclease protection assay, which just measured a variety of mRNA species for both cytoprotective and pro-apoptotic proteins. And the thing that really stood out, so everything here is at 4 and 12 weeks, and it's compared to baseline. The thing that really stood out was that the BCL2 mRNA levels were very high in these mice. And it was known that TNF could lead to degradation of BCL2 through the ubiquitin pathway. And so what we thought was perhaps that these very high levels of the mRNA were because BCL2 was turning over in the myocardium. And that was actually the case. So David Engel, who was a postdoc in the lab at that time, when we measured BCL2 protein levels in the myocardium at that time, you can see that they were reduced compared to the wild-type mice And then what David did was actually isolate mitochondria from isolated cardiac myocytes. And you can see that there was very little BCL2 protein that was in the mitochondria. If you remember that cartoon I showed you before, BCL2 is one of the proteins that stabilizes the mitochondria. So we thought that perhaps BCL2 loss through TNF activation was driving the apoptosis. And to get a better handle on that, what we did was to create a series of bi-transgenic mice. Michael Schneider, who was still at Baylor at that time, had developed mice that overexpressed human BCL2 in the myocardium. What we did was just simply marry his mice and our mice and created bi-transgenic mice. And you can see here that the BCL2 mice have robust levels of BCL2 in the cytosol and the mitochondria. There's very little, there's none in the TNF transgenic. Again, this is a human protein and we're using a human antibody. And you can see that in the bi-transgenic mice that there's some BCL2 in the cytosol, but a fair amount in the mitochondria. And because of this concept of promoter competition, anytime you use two promoters that have the same sequence, they may compete and drive down levels. We wanted to confirm that the TNF levels were similar in the myocardium, and they indeed were in the TNF transgenics and the TNF BCL2 mice. So we then went back and asked the question, how does this affect the cardiac remodeling process? So the first question we asked, does it knock down apoptosis? And it does. It does considerably, but the interesting thing was it didn't completely ablate it. We'll come back to that at the end. The next thing we wanted to do was to understand that cardiac remodeling process. And this is a busy slide, so I just ask you to focus in on the middle part here. So the TNF transgenics are shown in red. The bi-transgenic mice, actually this is mislabeled, are shown in green. And the interesting thing here is you can see that the TNF transgenics dilate, just like I showed you before. If you give the BCL2 or you create a bi-transgenic mice, you blunt the dilation a little bit, but not a lot. The really interesting finding here was that, again, you remember I showed you that MRI sequence where the TNF transgenics, the walls actually thin. And if you create a bi-transgenic mice that can't or has reduced apoptosis, the walls actually thicken. So we thought that one of the things that was driving the cardiac remodeling here was the cells dying and the walls actually thinning as the cells were dying. And again, if you look at the R to H ratio, it's actually increased in the TNF transgenics, and this ratio, which I said is normally invariant over time, was actually the same in the bi-transgenics at 4 and 12 weeks. So the kind of cardiac remodeling that was occurring here, one would consider as physiological. So to get at mechanism to this in the bi-transgenics, we again went back and did simple experiments where we looked at dumping the cytochrome C into the cytosol. And you can see, again, the bi-transgenics, it was attenuated but not completely abrogated. And so what we wanted to do was to try to begin to understand which of the cytoprotective pro-apoptotic molecules might be driving that. SMAC-Diablo is a so-called caspase-dependent pro-apoptotic factor that can drive apoptosis, largely by tying up proteins that prevent caspase-3 activation. And you can see here that in this, we were able to actually knock down SMAC-Diablo levels a little bit. And if one looked at AIF as a caspase-independent factor, it really had no effect on this, and we measured endo-G as well. So basically, the bi-transgenic mice had some decrease in the pro-apoptotic factors. The interesting thing is when you look at sort of anti-apoptotic factors such as CIAP1 and CIAP2, there was really no effect. And there's another protein called XIAP, and this was actually increased. And we think that this is largely through a proteasome problem, which I won't talk about today. But again, just to go back to the main theme here, very little effect on upregulating or changing pro-apoptotic or anti-apoptotic factors, but most of the pro-apoptotic factors were increased. So we went back and measured caspase-9 and 3, and you can see that they were partially but not completely attenuated in the mouse model. And this suggested to us that perhaps the other pathway, the so-called extrinsic pathway, might also be activated in the mouse model. So we went back and looked at markers of activation of the extrinsic pathway in the bi-transgenic mice. One would predict, since BCL2 acts down at the mitochondrial level, one wouldn't predict that there would be any change in the so-called extrinsic pathway, and that was actually indeed the case. We found evidence of activation of the extrinsic pathway in the TNF transgenics. Both caspase-8 activity was up, and there was cleavage a bit, and as one would predict, the BCL2 had no effect on it. So what we had done was block one apoptotic pathway, and the other one became activated and drove some of the cardiac remodeling. And we were interested in sort of understanding the mechanism for this, and if you'll remember the cartoon I showed you at the beginning, there's a molecule called C-flip, which ties up caspase-8 and prevents complex 2 from being activated, and that was actually degraded in the transgenic mice, as shown here. And it wasn't because there was actually less message. These proteins were actually being proteolytically cleaved. If you just do a ribonuclease protection assay, you can see there's actually more flip. So what does all this mean, and why am I boring you with all this? If you look at sort of how sustained TNF signaling drives the remodeling process through apoptosis, it's actually pretty complex. We think what happens is that there's primarily erosion of these so-called cytoprotective pathways, and that leads to activation of the intrinsic pathway. And in addition, there's subsequently erosion of the cytoprotective proteins that lead to block activation of the extrinsic. And so both of these pathways become activated over time, and I think that that's largely what's driving the increased prevalence of the apoptosis. So the results of the study suggest that, at least in vivo, apoptotic cell death doesn't occur as the direct result of activation of intrinsic or extrinsic cell death pathways, but rather cell death proceeds after sustained inflammatory signaling renders myocytes competent to die. In other words, once your reserve margin goes, then any bump at all will push you into apoptosis. And this depletion, we think, fosters a feed-forward amplifying mechanism that explains the increase in apoptosis. Now, why should anybody care about this? Well, if you're thinking about developing drugs to prevent remodeling, and you're thinking about identifying targets to do this, obviously one would not want to develop drug targets for apoptosis because the process is way, way too complex. You basically have to develop a pan-caspase inhibitor that inhibited all the caspase, and then you'd have people with cancers in about 2 to 3 years. So just to sort of reiterate what we've talked about so far, we've talked about the fact that tissue injury leads to matrix metalloproteinase activation. That leads to the dilatation, and we think that the apoptosis is largely responsible for the wall thinning. And both of these processes are happening at the same point in time. So where I'm going with this is that the biology of cardiac modeling, I think, is really extraordinarily complex, and it's driven by a number of changes shown on this slide. There are changes in the myocyte biology. They don't contract well. There's changes in myocyte cell number. They decrease. There are changes in volume of the complex of the extracellular matrix. You can't develop a matrix metalloproteinase inhibitor because if you do that early, that's good, but if you do it late, you get more fibrosis. And all the changes are dynamic, time-dependent, and interactive. And so I think it's likely that any attempts to interdict a single arm of the process will not be successful. And so this really summarized about 5 years of work in the lab, all to come up with there's really no magic bullet here to prevent cardiac remodeling. So over the years, this concept that the heart can heal. So what we try to do is say there's no one target that can prevent the thing from recovering, but what if one were to try to understand the process of recovery? Maybe that's the way to go through this. So I want to introduce this concept of myocardial recovery. I showed you data that if you give certain drugs or devices, the heart actually gets better. This was first observed in patients in whom we implant left ventricular assist devices. If you put these like little mini pumps in, the hearts actually get smaller. And in about 2 to 3% of the population, you can actually pull the pumps out and the heart failure gets all better. It was subsequently observed with all drugs and devices that improve patient outcomes. I showed you the data for that. Anatomically, this is characterized by a decrease in LV mass and a decreased volume, and if you measure pressure-volume relationships, they shift to the left. There's also a specific molecular signature for this, the so-called activation of the fetal gene program, which we'll talk about a little bit. Actually, they go back to normal. And the interesting thing here is that we don't understand a single thing about reverse remodeling. We don't understand any of we understand what drives remodeling forward, and I just showed you the data for that, but we have no clue about how a ventricle gets smaller. And if you think about it, is it that the myocyte contractility improves and then drags the matrix with it? Is it the matrix sort of gets better and then allows the myocyte to shorten more? We really don't know. And the other, I think, key question is there hysteresis? So after it dilates up and it comes back, does it come back to a complete level where you can take people off of drugs and devices and they don't need anything again, or does it come partially back where if you take them off the drugs and devices, the heart failure is going to get worse? So really where we're now focused in on the lab is, at least one part of the lab is understanding this process of reverse remodeling for myocardial recovery. And the reason we got into this was because of the development of these, our understanding of these microRNAs. But since coming here over the last 6 months, we've really begun to focus the lab on a systems biology approach. And I realize I'm talking to engineers, so I'm preaching to the converted probably on this. But one can imagine that the TNF transgenic models you could put here as your predictive model, and we know exactly how this thing remodels, and we've been able to parse it out, and we can measure it, and we know the biology of it. So we basically have predictive models. And what we're doing now is doing high throughput analysis both for microarrays of microRNAs and microarrays of mRNAs, and I'll talk about that in a second. And the idea would be to understand the key players that are driving this, not so much from a single approach, but from a network approach, and then be able to modulate that in systems and go back and measure it. And the reason why I showed you all the remodeling stuff is we can measure any part of this pathway now and tell you whether the microRNA you're putting in the system is impacting it in the way that one would like. So for those of you who aren't familiar with microRNA biology, these are small, non-coding microRNAs. What they do is they bind to target mRNAs through a seed sequence. They can either inhibit translation, which is the more common way, or they lead to message degradation. The interesting thing if you're a heart failure guy is that they're coordinately upregulated during stress. So there are specific patterns in microRNAs that come up under stress that we see, the so-called initial index event. They have predicted mRNA targets, so if you want to use a systems biology approach, you kind of know, if you know the microRNA, you know the genes involved. We've talked about the complexity of remodeling. I like microRNAs because they drive gene networks. We're not now talking about a specific pathway that a drug would hit. We now have a way to modulate an entire network. And as I'll show you on the next slide, they can influence cardiac remodeling. So this is a slide that I got from Eric Olson. So Rudy, you'll be happy that the microRNAs affect arrhythmias. Basically, these molecules can make anybody happy if you do experimental research. They can drive or inhibit apoptosis. They lead to angiogenesis, or they can block angiogenesis. They lead to cardiac myocyte hypertrophy, and they can lead to fibrosis. So basically, these are really cool molecules that impact the remodeling process, and they may have a way of actually leading to myocardial recovery. This is a slide from Eric Olson's lab that was published in Science. They identified miR-208 as a molecule that came up during pressure overload hypertrophy, and it was required for one of the fetal gene expressions. When the heart gets stressed, alpha myosin heavy chain protein goes down, beta myosin heavy chain protein goes up, and that's thought to lead to a slower contraction. And what they found was that, at least in cell models, is that miR-208 was responsible for beta myosin heavy chain expression, which is required for cardiac hypertrophy and fibrosis. So when they knocked it out and then pressure overloaded the ventricles, you can see that the mouse develops a heart failure phenotype in the wild type. And if you pressure overload that mouse but knock miR-208 out, they don't remodel. So you can begin to see the power of these molecules. So we've been interested in trying to use miR technology as a way of studying both remodeling and reverse remodeling of myocardial recovery, but what we needed to do was to develop a better mouse model because the TNF transgenic is very dirty. It activates a variety of pathways that you would rather not activate if you just want to study remodeling. So we've focused largely on a molecule called TRAF2, which is downstream of both the type 1 receptor and the type 2 receptor for TNF signaling. And the reason we got interested in this is that it activates pathways that we think are really important for cardiac remodeling. The NF-kappa B pathway, as well as the AP1 pathway, which is linked into cardiac hypertrophy. So what we did as a first pass was just create a TNF transgenic that overexpressed TRAF2. We presented this last year. the American Heart Association, and we just asked the question, does it phenocopy the TNF transgenic? Is this the pathway that we want to focus in on and develop a mouse model? And the answer is yes. They basically develop adverse remodeling, depressed LV function. They have increased MMP activity, increased TIMP level, increased apoptosis, and increased NF-kappa B activation. So we think that the molecules that are driving cardiac remodeling are downstream from TRAF2. And so the real question is then, could you then use this system, which is a little bit purer for understanding remodeling, if you were able to create a bitransgenic mouse system, so if you're able to turn TRAF2 on and able to turn TRAF2 off, and then you could then study remodeling as it goes forward, and you could then study remodeling when it goes back. Would this be a way to sort of uncover novel targets for developing therapies that allow the heart to heal? This is a system that was developed by Jeff Robbins. It's a traditional binary transgenic model. What they do is drive a TTA activator through the alpha myosin heavy chain promoter, and then what they do is they have a responder protein that you can then insert the transgene of interest. In our case, it was TRAF2, and this is a TET-off system. So in the presence of tetracycline, TET will bind to the TT activator and prevent it from activating the downstream transgene. If tetracycline or doxycycline is not present, then the TTA activator will then bind to the TET operon, and the BP16 will then drive expression of the TRAF2. So all we did was to create transgenic mice that we actually got these from Jeff Robbins. We then created a transgene with TRAF2 down from the responder construct. We then marry these mice, and unfortunately, if you want to be a purist about controls, and I tend to be, you actually have to have three different controls here. So you have your wild type, you have your TTA mice, and your TRAF2 mice. And these really, this mouse here is only this construct here. This mouse is this construct here. Neither of these will express the transgene. When you marry these together, in the absence of tetracycline or doxycycline, the transgene will activate. And then when you give dox, you can turn it off. And so here we've just shown in panel D that the system's really pretty responsive. So these are mice that were allowed to go 6 weeks, the so-called dox off. We then give doxycycline, and you can see that the transgene turns off relatively quickly. Within 2 weeks, there's completely turned off. And the nice part about this system is it really doesn't leak. So we've now got a system that we can measure forward remodeling and reverse remodeling, and we're still finishing these studies. But the basic concept is we take wild-type mice and then the TTA TRAF2 mice. You either get diluent, no dox, or they get dox. And we determined that at 8 weeks, the hearts will remodel, and then from 8 to 12, we can actually drive the whole process backwards by giving doxycycline. So we have a mini clinical trial here. And then these are all the standard things that one would measure that I showed you before. So there's no magic to the endpoints here. So this is what the mice look like if at 8 weeks of age they develop increased heart weight to body weight ratio. They actually begin to dilate and they get cardiac hypertrophy. And you can see that if you give the doxycycline from 8 to 12 weeks, that you can prevent, or actually not prevent, but you actually drive it backwards. So these hearts are dilated and now you're driving it backwards by deactivating the gene. And if you just give them diluent, they continue to dilate. If one then looks at the things that I talked about before, they dilate, as I said before. The wall thickness doesn't change much because there's not a lot of apoptosis in this mouse. But the R to H ratio, which I showed you before, are the fractional shortening. So the contractility is going down in the mice, at least in vivo, and the R to H ratio, which I showed you before, goes up. And if you turn the transgene off, it all goes back to normal. If one measures LV function on a Langendorf apparatus, so we take the hearts out, perfuse them with buffer, and just measure contractility, completely absent of the adrenergic nervous system, the developed pressure in the TTA mice is not very, is depressed. And this actually returns to normal. I'll show you in the next slide if you turn it off. There's not much effect on diastolic function, so this was a little bit of a disappointment. But maybe if we let the mice go longer, we'll get more diastolic dysfunction. The interesting thing here, if you look at the mice, if you from 8 to 12 weeks of age, if you don't give them any doxycycline, they have depressed function. You can completely reverse the contractile defect here by giving the mice doxycycline. And again, there's really no effect on no real effect on diastolic function. You were talking about Steve Hauser showed us how to isolate myocytes. So we have been able to isolate cardiac myocytes from these mouse models in both the wild type that was given doxycycline, the TTA-TRAF2 mice that developed contractile function in the absence of doxycycline, and then the TTA-TRAF2 mice that are given doxycycline. So this measures the sarcomere shortening in individual cardiac myocytes. And you can see that they develop contractile dysfunction that's largely but not completely reversible after you give the doxycycline for 4 weeks of age. Again, these are just group data here showing that there's depressed function at 12 and that you can completely reverse it. And we talked a little bit about fetal gene expression. So there's a molecule known as ANP, atrial natriuretic peptide, and ENP, E-type natriuretic peptide. Both of these are signature genes for fetal gene program. In the heart failure model, they activate, and you can actually turn the transgene off by giving the doxycycline. Alpha myosin heavy chain, which we talked before, actually goes down. In the model develops heart failure and goes back up. In the mice given doxycycline, not significant. We have to do a few more mice. And then the beta myosin heavy chain, which goes up, is actually reversed. So there's a partial reversion of the fetal gene program. So what I've done is really show you that we've got, I think, a reasonable heart failure model that undergoes recovery. And so the real question is, what's driving the whole recovery process? So the strategy we've taken, and we're in the middle of doing, is to identify microRNAs that are upregulated during cardiac remodeling and downregulated during reverse remodeling, and then study those microRNAs whose predicted gene targets are known to be involved in cardiac remodeling. So I said we kind of mapped out the whole remodeling process over 5 years, so we know the pathways that we think are important. So the strategy that we've undertaken in Nelly Topkara, who's the postdoc on this, we've identified 43 miRs that go up during remodeling and reverse during myocardial recovery, and 29 miRs that are going down and going up. The initial plan was to only study this group, but we've just decided we're going to take anything that changes in a mirror image of itself is what we're going to put into a predictive model that we're developing with Charles Gu. And what we're also doing now is extensively categorizing all the mRNAs. So we've done microarrays of all the microRNAs, and microRNAs of all the genes that are involved in this. And the plan here really is to try to develop a model with Charles Gu, and we've actually got an ad out for a postdoc to help do this, because it's going to require a lot of computer modeling. But the idea here is to take the microRNAs of interest. I showed you we've got about 60 of these things. Each of them has probably about 100 predicted gene targets. So with the computer, or about 18 postdocs, and I notice you have about 12 in your lab, you can actually come up with all the predicted targets for the microRNAs of interest. What we want to do is then screen these targets using actual lab data of the mRNAs that are changing in a direction. It doesn't matter whether they're going up or down. The only prerequisite we have is that they're actually going up. And then the interesting part is, in the literature, we know from these left ventricular assist devices, in humans, we actually know the genes that go up and go down with LVADs. So rather than looking at 10,000 genes, we're able probably to come up with maybe 1,000 genes that look interesting. And then because we know the pathways that are involved in remodeling, the reason I showed you all these different things, is we can now project these gene targets on various aspects of the remodeling process that we think are important. And the idea here would be to come up with some candidate microRNAs and then put these back into the mouse models of cardiac injury, either blocking the microRNAs through the use of antagomeres, or if we think that the mirror going down is leading to activation of adverse genes, we can also give mirror mimics. And my prediction, if you believe network biology, is that it won't be a single mirror. You're going to probably have to use cocktails of these. But the nice thing is, because we've got mouse models that we can do this, and the mouse model really develops over about 8 to 12 weeks of age, you can actually inject these mirrors through a tail vein injection, and we can then go back and remodel, or look at how it affects remodeling. And the idea would be to develop ways of manipulating networks that we might be able to take in humans. And if you think that this is all Mr. Wizard stuff, actually Eric Olson's got a company called Miragen, and they're taking not not quite this approach, but they've identified specific miRs and they're trying to get them into people. They've actually been able to encapsulate them in lipid endosomes and get them into cells to drive it. So there's actually a company that's doing it, and just by way of disclosure, I'm on the scientific advisory board. So I got to thank the people who do the work, because I do very little these days. Natarajan Subramanian stayed in Houston. Unfortunately, he was instrumental in developing the early transgenics. DJ Devakaran did the work with the TRAF2. Feng Gao and Wei Wang were both incredibly talented investigators in the lab. David Engel did the work with apoptosis. Sandra, she's now assistant professor at Baylor. He's assistant professor at Columbia. The group that we've assembled here at WashU, Phil Barker, who followed me from Houston, is sort of helping to run the lab. Weili Zhang, who's in the audience here, did some of the work with fibrosis. Hui Ping has done some of the work with the TNF transgenics, and a lot of the work with the miRs has been driven by LV Remodeling. And we've established a preliminary collaboration with Charles Hu, who I hope can help us with all the bioinformatics, because there probably aren't enough postdocs in St. Louis to handle all the gene targets. But the approach we're taking, I think, will work. There's actually a paper in JVC published from the Cleveland Clinic Group that's done a very similar kind of modeling approach to this. They're only looking at forward remodeling. So I want to return to the chicken and the egg question, because I promised you I would do this. And so many of you in the audience are probably still wondering whether it's cardiac remodeling is a cause or effect for heart failure. The chicken here, as I said, still can't make up his or her mind. Those of you who are very sharp will notice that the egg is in the shape of a prolatal ellipse, which is sort of how the normal ventricle is. So the egg thinks that the geometry is destiny, but having come from the great state of Texas, and you guys are engineers, so you would definitely concur that what really matters is the size. So thank you very much. I'd be happy to answer any questions. Now I have to finish my coffee.
Thank you for a fantastic talk. So, questions to Dr. Mann?
Yeah, Tom. So I want your Mac back.
Yeah. I've seen some talk about these little nets that you put around the heart that sort of size.
So I was the principal investigator on that study, which I didn't show.
What are your thoughts on it?
Yeah, so there are a lot of ways to do this. Our first approach, there was what Tom's referring to is there was a filamentous yarn that could be wrapped around the heart. And when you do that, it actually unloads the heart. It exerts about 2 millimeters of pressure on the heart. And if you look at what drives the calculation of wall stress, it's the so-called distending pressure. So just by wrapping this around, you actually decrease the transmural pressure, you decrease wall stress. We did a clinical trial with this device in about 300 people. The primary endpoint of the trial was a composite of people getting worse or getting better from quality of life and not needing major cardiac procedures. And the major endpoint of the trial was hit. The FDA had problems with, we had some early mortality. There were two arms. One was the device was put on people who were getting microregurgitation, surgery, MBRs, replacements or repairs. And the other group was just purely medical. And there were deaths in the early arm, primarily in the big ventricles. So although the primary endpoint of the trial was met, the FDA had safety concerns and then wanted to do a second study. And the really dirty secret of developing drugs and devices for heart failure is that it costs like millions and millions and millions of dollars. And the company kind of got sideways with the FDA, tried to sue them, force it through the process. And so basically the FDA killed the whole project. And the company, they couldn't raise money because of the nasty sort of financial climate. So the initial approach was, again, I've shown you how complex remodeling is. So today, with all we know about cardiac arrhythmias, what do? We put a box in people to prevent the arrhythmias. Because I grew up giving all these drugs and measuring PVCs and counting them, and that didn't do any good. So I still think that the best way to do this would be a mechanical way that maybe either had cells embedded in it that might elute microRNAs that decrease fibrosis. There are clever ways to do it. But the so-called Acorn device didn't get FDA approval, and now the whole thing is just so complex with loss of funding and antipathy with the FDA that I don't think we're going to get the device. I think the idea is right.
Yeah.
Somebody once said, how can you tell the explorers they're the ones with the arrows in their back? So I've been pulling arrows out of my back for about 2 years on that one. But we just couldn't get it past the FDA, although the principle, I think, is right. We just couldn't demonstrate the safety of the device, and the company didn't have the money to go do a confirmatory trial. So, yeah.
Regarding the reverse remodeling system at the end, do you think that could be used as a way of sort of extracting important events that happen that prevent reverse remodeling? In other words, what would be a characteristic that would happen at a longer time point that might prevent the regression?
So that's a great question. So I didn't show the data because I realized I didn't want to bore too many people too much more. If you let the model go out too far, it doesn't come back all the way. And that goes to my comment about hysteresis. So with this, not only will we be able to sort of try to understand what are the basic movers that make the heart get smaller. When the heart gets better, what's driving it? That's really the question that the lab's interested in. The other thing is what are the determinants of irreversibility? Today we spend $50 million on just heart failure therapies alone, and we're probably giving devices and drugs to people who are never going to get better. And sometimes it sounds cold to say, but maybe hospice care. I mean, sometimes it's okay for people to die, and I think we're spending a lot of money on people who aren't going to get better. And you can also get the determinants of irreversibility. I think using these two, we'll be able to figure out, once certain networks are activated, can you not come back from it? And again, that may be a way to prevent things from getting worse. So I think the model system should allow us to do a variety of different things. The real limitation is that it's strictly driven by inflammation. We want to collaborate with other people and take a couple other different models that develop the same phenotype and then express them in an inducible manner and see if we can look at common genes that are common to both. The TRAF2 pathway, let's say an adrenergic pathway, or one that disrupts cytoskeleton or calcium handling or something. So the weakness here is that you only have one model system, and obviously heart failure is a very heterogeneous disease. So the idea would be, if we can get the system that Charles do, where we can actually dial in things and understand it, we want to create 2 to 3 other models, and then look at the commonality of genes. And you can only do this with computer modeling. It's just way, way too complex because I burned out the poor postdoc. Looking at setting up the genes on Excel spreadsheets, it just won't work. We have to get a way to get it up on a computer and do the modeling. But yeah.
I was curious, why not try to block the action of TNF alpha with something like Enbrel? And the second part to that question is why is heart failure a counterindication for your patients then?
Yeah. So the one arrow on my back is from the Acorn device. I was the principal investigator on the Enbrel trial too, and we'd actually, that was something we developed in a culture dish, took it all the way to a phase 3 trial. The trial was negative. It was about 3,000 people conducted in North America and Europe. And again, this goes with the politics of drug development. We went from phase 1 in 60 patients. So we did it in a culture dish, we did it in a rat, and then we did it in 60 patients. And then they went to an international trial. Too quick, in my view. But the company was worried that if they didn't hit it, they would be bought out. And today, they're called Amgen, I think, bought them out. So Immunex got bought out by Amgen. But the trials were negative when we gave etanercept. And because of the futility analysis, it was really hard to figure out why the drug didn't work. In North America, there was actually worsening of heart failure, which is where the black box came from. In Europe, people were only on it for 6 months, and there was a little bit of an efficacy signal. Now, Enbrel is a funny drug. What are you referring to? Enbrel is 2 of the TNF receptors, the type 2 TNF receptors, fused in duplicate to the IgG1 molecule. And they do that to keep it in the circulation. When Enbrel was first sort of engineered, they made this observation that at certain concentrations of TNF and Enbrel, that actually it acted as an agonist. And the reason it acted as an agonist is that when TNF binds to it, it assembles it. TNF is normally a monomer in the circulation, assembles as a homotrimer, and then it's biologically active. Well, it would bind to etanercept, etanercept would stabilize it, and then give it back to the cell. So if you look at the original paper by Moeller, where they did the patent on that, they actually made this comment that at certain concentrations it's an agonist. We did subsequent modeling, just published it last year in the Journal of Clinical Translational Science, and showed a similar type thing. At a specific ratio of TNF to etanercept, etanercept can act as an agonist and actually increases TNF bioactivity. And we think it's because it stabilizes TNF and it gives it back to the receptors. The reason why I don't think you have trouble in rheumatoid arthritis is that the TNF levels in heart failure are very high. In rheumatoid arthritis, they're relatively low. And so the amount of increase you get with rheumatoid arthritis is from here to here, and what happens in heart failure doubles. So if you start with a very low baseline, the amount of agonistic activity is small because there's not much TNF to stabilize. If you start with a lot of TNF and then stabilize it, it gets worse. And you can actually show almost a twofold increase in cell killing. So one explanation for why it didn't work, and that's the one that I do when I'm feeling cowardly, is that the drug caused harm and caused worsening heart failure because of the agonistic properties. And if you look very carefully in the paper, in the group, one group that received the drug the longest actually had worsening heart failure. It was statistically significant. So that's why. There was another trial with infliximab, which I really cautioned the company not to do. So infliximab is a humanized antibody that has a murine, it was genetically engineered so it binds TNF, but it's an antibody. And so I showed you the pictures where TNF is expressed in the heart. If you think about it, an antibody with an Fc portion on it is going to home to the myocardium, bind to TNF on myocytes, and then lyse them when you get activation of complement. And that's exactly what they had. They gave infliximab to these patients, and there was like a 4- or 5-fold increase in death and hospitalization in the patients who were given infliximab. So they got a black box on that. But so we initially tried the pathway of blocking TNF, and it didn't work. And then we tried a variety of other things, thalidomides. So then we went to broad-based molecules that had a broad spectrum of action, like thalidomide. Thalidomide was the drug that was used as a sedative. Then they found that it had teratogenic properties, which was basically causing a lot of malformed kids. So it got taken off the market, but it's a great anti-inflammatory, and they use it in multiple myeloma. So we had a very effective, it actually made people feel a little bit better, but at the high doses, which is what it took to knock TNF down, I put everybody to sleep. So that got killed. And then there were a couple of other things. And after that, I kind of decided that I wasn't as smart as I thought I was, and that you can't move that quickly, and you got to go back to biology. But you did the etanercept thing, and then when the device with Acorn came out, I thought that was really good. And that was frustrating because it actually worked, but we just couldn't convince the FDA to let it. So it's not as easy as it seems. And actually, it's amazing we have any drugs that work at all today. I have tremendous respect for the pharmaceutical industry because it's really hard to develop drugs. But the etanercept thing seemed too easy, right? And we did it, got the FDA, not the FDA, but the NIH to fund it as part of a SCORE grant. And as I said, the mistake I think we made was going from 60 people to 3,000 people. You really need to have phase 2 development, drug development, to begin to see complications. And those are all the arrows I have. I only have 2 sets of arrows in my back. You can ask any other question you want.
Any nice question?
I have a load question, if I could. So lots of times in these heart failure patients, what happens is their ventricular enlargements get hypertensive. And one can't help but realize that in many of the people who get better and the heart shrinks, their mean arterial pressure and their load also goes down. So this is partially a chicken and the egg question, which is after the onset of the initial injury and the afterload goes up, are you looking at an epiphenomenon regarding that? And if all you need to do is control the blood pressure, the mean arterial pressure, all the positive remodeling will just follow on its own.
Yeah, so at least in heart failure, excessive load is usually not a problem because the blood pressures or SVRs can be high. But and relative to the contractile ability of the ventricle, it's probably an afterload mismatch. My own view is that the mechanical burdens imposed by cardiac remodeling with the increased wall stress probably far outstrips the problem of increased afterload average blood pressure in a patient in my clinic when I was seeing a lot of heart failure patients was about 80.
Those were
Yeah.
that's after the capacitor mechanisms fail. And those aren't the ones that remodel. Let me say it another way.
Okay.
Is there evidence that people who have heart failure and who are hypertensive, if you keep their pressures high, you can get their ventricles to shrink? In other words,
I'm unaware
possibly
of any evidence. I can tell you that the people who have hypertension and heart failure have a better prognosis. But that probably is because the ventricle's got enough steam left in it to generate hypertension. But if you look at sort of prognostic factors, a bad prognostic factor for heart failure is a relatively low blood pressure. Good prognostic factors. If you can muster 140/80, that's a really good thing. Yes, sir.
What do you think of the various efforts to use stem cells to treat heart failure?
I'm new here, so I don't know if there are any stem cell people in the audience. I don't want to offend. Any stem cell people in the audience? Chandra, are they being honest?
Yes.
Yeah, okay. So, first of all, it's fascinating biology, and I think the studies should be done. I have been negative about stem cells for a couple of reasons. One, if the initial attempts were all through injection. I kind of was trained in the San Diego environment where all we did was study ventricular mechanics. There are 7 layers of the myocardium. They're all sort of interlacing in different ways, and it's amazingly complex. So imagine injecting a stem cell in there without blood supply, trying to get it to organize in the right direction, not go sideways, then get it to intercalate, develop an intercalated disc, and still contract. The chances of that happening are slim to none. Okay, so the injection stuff I never liked. Subsequent studies by Victor Zhao's group have shown that a lot of the effects that we're seeing, the beneficial effects, at least in mouse models, are probably driven by paracrine factors. That I believe in. Okay, so I think that another group of the lab is studying sort of innate stress responses, and I believe when cells die or are in the process of dying, they do release the so-called endokines or so-called alarmins, the other thing they've been called, that can then activate cytoprotective signaling pathways. So, but I think that's a fundamental property of all cells, biological property of cells, that they are able to tell their neighboring cells, I'm in trouble, upregulate your cytoprotective pathways. That probably has very little to do with stem cells. I mean, the whole idea with stem cells is that they were going to differentiate into myocytes, and the data on that are really very slim. I mean, the frequency with which it happens is hotly debated, and some people insist it doesn't happen, some people insist that it happens a lot, but really you can't get anybody to agree. If you just take a step back from the biology of it and say, we don't understand the biology, but there's some signal, and you try to translate that now to a clinical trial, it becomes hard. So which cell type are you going to use? The basic guys can't agree on how the things work, so how are you going to select a drug to go forward into a trial of 10,000 people, which is what I estimated it would take to show a mortality benefit. And then the mode of injection, when you inject it down the coronaries, which is the easiest way. They've done some really nice studies showing that the cell, I think, resides in the myocardium for a couple hours and then either moves on, dies, or something happens. So then you got drug delivery problems. I've already articulated that I don't think direct myocardial injections are feasible. So it becomes very hard to design a clinical trial that would show a morbidity and mortality benefit. All of the results that have looked good so far have been from ejection fraction changes, and they're like on the 3 to 5% change, and that's within the era of measurement. And then there's only one study that's looked at long-term follow-up, so they found that if you give stem cells, the EF goes up 5%, and then in 6 months the control group catches up. And then the real problem I have with all of it is that I believe in this remodeling thing. All the effects have been shown. There's no really effect on remodeling, which every single device or drug that saves lives or makes people feel better has some impact on the remodeling process. And so all the studies that have looked at this have not been able to show it. So I think the biology is cool. I think that there's really something there, but I don't think it has much to do with stem cells. I think it has a lot to do with basic cell properties. And if you think about it, when cells die and you have an infarct or a heart attack, there's healing that goes on in the myocardium. And I think all these cells that we're putting in are really accentuating or accelerating the healing process. So I think that I have a lot of, I believe in. But but I don't think it has much to do with stem cells. But again, I thought the Acorn device was going to work. I thought the Tanacept was going to work, so I'm probably the last guy. I also predict ER would only last one season on TV. And my track record so far is not really good, but I think there's something to the biology. I just don't think that it's the way that they're doing it now is going to pan out. And the real fear I have is that everybody's moving so fast and has so many different companies that you're going to get a couple negative results and they'll kill the whole field, and then everybody will walk away from it and won't be able to do the really important studies. We move too fast, I think, because there's too much money involved, and a lot of times it's driven by corporate
Companies run out of money.
Yeah, companies run out of money, and that's why Immunex went after this. They saw it as a way to become independent, much bigger market than moving toward arthritis. And despite the protestations of myself and a number of other people, they just went ahead and did the study, and then it bankrupted the company. You watched their stock went from 10 billion to 5 billion in one day. It was wiped them out, all because anyway, so hopefully I haven't irritated too many stem cells.
So you've identified multitude of targets on microRNA and mRNA changed in this mouse model during forward and reverse remodeling. But do we have any data showing what the fraction of all these targets would also correspond to similar remodeling in human disease?
Yes, so if you
What is the
what
percentage?
we're trying to do so that we're sensitive to this is there are probably about 5 or 6 studies that have been published where they look at these mRNA changes in patients who've had these left ventricular assist devices. So those of you who don't know, we're able to put small mini pumps in the apex. The beauty of that is you get a core of ventricular tissue at the time you implant. You unload the heart. About 3 to 5% of the people actually get all better and can come off the pump. Most of them go to transplant. But at the time of transplant, after the heart's been unloaded, you can then get the ventricle, as you know, and look at that. So there are about 5 studies that have published full mRNAs. Gerald Dorn has one study on microRNAs, but if you read the paper carefully, it's not paired. Okay, they didn't look at paired samples. If you want to be a purist, I would argue you should look at before and after the way we can do. So what we're trying to do as one of our filters is also use human gene targets, because I believe that we study a lot of mice, but a lot of times it doesn't translate to humans. So we're not only using experimental data, we want to use published data out available on the web to help us screen our first candidate targets to predict the microRNA. So I completely agree with you about the importance of human physiology or neglect of it in many times. And that's why, as I said, we've kind of built that in as necessary, another screen to help us at least focus in on the genes that we think are important.
But there's another complication to that, like in the arrhythmia field, we have a lot of data from atria, for example, from where we can get it, which is atrial appendage.
Yeah.
Well, we know that heterogeneity expression is so significant that it might not be represented. So similarly here, you get tissue from one region, which happen to be next to the device, which perhaps remodels in a completely different fashion.
Yeah. Again, one thing you could also argue is that the platforms that have been used and the tissue processing that's used hasn't been pristine. So one of the other things I want to spend some of my startup package on is getting LVAD samples here that we collect using your protocols in collaboration with you, and get really pristine samples, and then do sort of high sensitivity mRNA analysis again to see if we get the literature that matches. It's difficult, in this environment. For example, patient X may get an inotrope, patient B may not get an inotrope. One patient may have an acute episode of ischemia before the LVAD comes out, another one may not. This isn't as simple as it seems, but in parsing it out between epicardium, endocardium, and mid-wall, I'd be happy to try to look at those samples, we could do, but my suspicion is there's going to be a lot of noise. We don't even know, what drives. Does the endocardium have to get better? Does the mid-wall have to get better? And the epicardium, which one happens first? Very, very complex. I'll be happy if we can identify 5 to 10 microRNAs, figure out a way to put them in a tail vein injection, and then at least measure something in injury models to see what this obviously complex. The good news is that there's lots to do. Need more arrows from my back. Yes?
Can you elaborate on the idea you mentioned where you implant cells that elute the microRNAs?
Yeah, so there's a lot of really interesting stuff going on now with tissue engineering for the heart. The initial concept is, you know, we can grow new hearts. If you think about it, that's hard. If you think, I didn't show any of the slides with the acorn device, but there are also sort of dacron grafts and weaves that people have implanted over parts of the heart that are infarct. And they've shown that if you just do that, it prevents remodeling. So one of the things I've wanted to do is actually collaborate with some bioengineering types who would be interested in getting fiber meshes, implanting cells on there. It really almost doesn't matter the cell type as long as you can genetically engineer them. But you could, let's say you wanted to get something that knocked down fibrosis. We just identified as year 29B, Olson's Labs 29A, as being microRNAs that prevent cardiac fibrosis. You could ostensibly get a mesh. The surgeon can put it on at the time they're doing the bypass. You implant the mesh over the area of tissue injury. You seed it with the cells, and you can do it in an inducible manner if you want. You could turn the cells on to secrete microRNAs of interest to drive networks that were antifibrotic or that caused hypertrophy. You can imagine all different kinds, or if you thought, you know, angiogenesis was your target. So you can have unlimited targets, but there's really good evidence, at least in animal models, that just a simple mechanical patch prevents a lot of the remodeling. And I think, you know, if you think 2 steps forward, you could seed that patch with biological material and then engineer it to secrete the things that you want to. Again, you'd have to pick your targets carefully, and then come up with ways of seeing whether it affects, you know, the remodeling process. The nice thing is that there are readouts that one can use for this, and so you can actually measure. You do it, and you can do it in people, and then you can measure people. You can do the same thing with the LVADs. You can actually, one of the things I'm trying to convince Olson's group to do and to fund, you can actually, at the time you put in the LVAD, you can inject your microRNAs and then dilute it on opposite sides of the ventricle, and then you can, at the time you take the heart out, you can actually see whether these things are doing what you think they do in a human, so you can actually measure them. You can measure ion channel activation too. So there's a lot of very cool stuff that's right at the nexus between cell biology and plain old engineering. Not plain old engineering is never plain old, right? But engineering and then biology, where you could use these types of things. So I think it's an exciting time for young people to get involved in this. And translational medicine is a lot of fun. It's frustrating because you don't always hit a home run. Sometimes you get hit by the pitch, but it's a lot of fun. So I think that's an opportunity too. I don't know if we have good tissue engineering people here at WashU, but it'd be the kind of thing to do.
There are good people in this department. Any other questions or comments? Well, thank you so much for coming in.